Good evening, students. This is the next chapter, chapter number 5. As you see, after the principle of inheritance, there is one new chapter, that is molecular basis of inheritance. So, we have to start this chapter right now. Actually, molecular basis is depend on the genetic, and the genetic is depend on the nucleic acids. Two type of the nucleic acid is there. One is a RNA, another is a DNA. But we don't know which one is the best genetic material. Actually, we know, but according to this chapter, we don't know. So, this chapter tells us which is the real genetic material. So, before that, genetic material is somewhat which contain a genetic information. Actually, it is a type of a material which contain a genetic information, and the genetic material is of two types. As we see, the nucleic acid is of two types, but genetic material is of two types. One is a DNA, another is a RNA. So we have to prove by theoretically and experimentally how the DNA is a best genetic material. First of all, if we go for the theoretical purpose, so first point say about it, it is a polymer of a deoxyribose sugar. Deoxyribose means ribose sugar is there, but D means double. So double ribose sugar is there because it has a two strand. It is present in all living cell, can replicate, have a specific type of a sequence, able to repair itself, and it is very sensitive to the environment. Experimentally, there are two type of the experiment happened at the time to prove that DNA is actually a genetic material. The first one is the Griffith experiment, and the second one is Hershey and Chase experiment. Griffith actually work on the mice, चूहे के ऊपर. So you take the two type of a bacteria, one is a virulent, another is non-virulent. So the process is going on. First of all, he injected the non-virulent bacteria in a mice. Somehow the mice survived. But when he injected the virulent bacteria, the mice died. And somehow the experiment is going on, and further the virulent bacteria is going to be heated. As you already know, bacteria become inactive during the time of the heat, and so the solution is going to be heated, and because of that the mice died. But when this violent heated material is combined with a non-violent, as you see, the non-violent mice survived also, and the violent heated, that also the mice survived. But when it is mixed, the expected result must be mice can survive. But the result which is coming after the observation is that mice died. So he experimented that there is somehow present inside the virulent bacteria. When we heat the virulent bacteria, it transferred to the non-virulent bacteria and become virulent. So that material goes to the another non-virulent bacteria, and because of that, that non-virulent bacteria become virulent, and because of that the mice died. So it is due to some genetic material. The second experiment is that Hershey and Chase taken the bacteriophage. As you see, the bacteriophage break down the cell wall of a bacteria, and he also take the E. coli bacteria. But here he do the experiment in the two phases. He using the radioactive sulfur. Firstly, he used the radioactive sulfur on the protein coat of bacteriophage, outer cover of bacteriophage, and inject the bacteriophage inside the bacteria manually. Because of that, the bacteriophage increases its population inside that E. coli bacteria, and when the new progeny is going to be formed, there is no sign of a radioactivity in the next generation. So it shows that the protein cover, which is outside of that protein, outside that bacteriophage, is not a genetic material. But when this bacteriophage consists of inside material is radio-labeled by the help of the sulfur that inject in the host, and in the next generation the progeny also consists of the same type of the radio labeled sulfur DNA. This says it is due to the DNA, and it is proved that the genetic material which is transferred, which is the double standard circular DNA, is actually transferred to the another generation. So one thing is this one. Now the next one is structure of a DNA. Structure of the DNA said that DNA is a double helical in structure, discovered by the Watson and Crick in 1953. Erwin Chargaff rule said that adenine thymine is equal to the concentration of guanine and cytosine constant. The total percentage is like that. If I can say, if the 30%, if total the 100%, the adenine is 30%, so thymine is also 30%. And 30-30 means 60% is adenine and thymine. So we can say guanine and cytosine is now 40%. Now it consists of two polynucleotide chain of a sugar phosphate backbone, and also the polarity is from 3:5 and 5:3. There are two strands there. The bonding between the base pair is hydrogen bond. Adenine combined with the thymine with double bond, and guanine with the cytosine by triple bond. The chains are coiled in a right bond direction, and the distance between two base pair is 0.34 nanometer. Each turn consists of 10 base pairs, so the distance is total 10 into 0.234 becomes 3.4 nanometer. Enzymes which are used in this are helicase, polymerase, ligase and primase. Even it has a 5 type of a histone protein, H1, H2A, H2B, H3 and H4, which combined and form the octamer. Now the next thing is DNA replication. Actually DNA make the copy of itself and taken the 7 steps to make a copy. The first step is the recognition of an initiation point. The second one is unwind itself. Then it create a template strand. Then the use of RNA primer. After that the elongation purpose is happening and replication fork is going on, and the last one proofreading. So in the initiation point, total 100 to 200 base pair of nucleotide must be taken at the initiation point, starting point. Specific initiation protein needed to specify where the initiation point actually is. In prokaryotic, there is one initiation point, but in eukaryotic cell, there are many initiation points. For the unwinding of the DNA, helicase unwind the part of a strand. Now if you go for the template DNA, the single standard, now the DNA becomes single standard. One strand another way and there is another direction. So single standard DNA is go for the synthesis purpose. Then we go for the RNA primer. Actually this RNA primer is a RNA polymerase, which is a preformed type of a nucleotide sequence, a small type of the fragments of a nucleotide which are bind to this DNA and show the way of RNA polymerase to find out where is the template DNA and from where it has to go on the process. The next one is elongation, where the DNA polymerase 3 synthesized new strand from 5:3 by polymerizing process. The process is actually called as a polymerizing process. And the replication fork, which is formed like as a chain, open the DNA into two type of the things. One is a discontinued pattern, another is a continued pattern. So somehow the question will arise is, what is discontinued pattern? The discontinued pattern is actually 5:3, which is called as a Okazaki fragment, also called as the lagging strand. If we go for the continuous pattern, it is 3:5, which is called as the continuous strand and leading strand. But in lagging strand, the lag is because of the gaps, and these gaps are filled and joined by the help of an enzyme called as a ligase, as I already show you here. Now for the proofreading purpose, DNA polymerase 1 and DNA polymerase 3 used to go along the new strand which are formed and read, and if any mistake is going to be formed, then or find out any type of the mistake is happening, then it can be corrected. This is the total DNA replication, also called as a DNA duplication. The second one, fourth part is a DNA transcription. DNA transcription is actually from the formation of RNA, that is mRNA from DNA. Has a 3 stages. Number 1 initiation, number 2 elongation, number 3 termination. So go for the initiation. Initiation actually consists of RNA polymerase, We have to form the RNA. So here polymerase is RNA polymerase, actually joined with the promoter, and there are some transcription factor, means transcription protein also added to it. So the first bond is formed, and then the promoter will release. As the first form is, जैसे ही first bond form होता है, the promoter will release and leave the place. And to leave the RNA polymerase, at least 23 nucleotide must be transcribed. जब तक 23 न्यूक्लियोटाइड ट्रांसक्राइब नहीं होती हैं, RNA polymerase will not leave the place, and then RNA polymerase leave. In the elongation purpose, one strand used as a template strand. Promoter recognize the sequence and many copies are going to be formed, but actually when the termination is going to be happen, polyadenylation occurs. So polyadenylation actually occurs in termination. At this stage, polyadenylation means the addition of poly A tail. Adenine is going to be joined here. So a series of poly A tail is added, and because of this, when this AAA is going to be formed, it is the tail end. At this end, mRNA is formed and left the part of there where it is going to be formed. So this is the DNA transcription. Now our next part is RNA and its types. Listen it very carefully. First of all, RNA. RNA is actually a single standard. It also have a adenine, guanine, cytosine and uracil. First of all, it is responsible for the coding, decoding purpose, regulation purpose, expression purpose of a gene. In the fifth end of the RNA, phosphate group is there, and in the third end of the RNA, hydroxyl group is there. If we go for the types, there are three types of RNA: tRNA, rRNA, mRNA. Actually, this tRNA is 75 to 95 base pair long. Transfer the amino acids and have a specific tRNA. Each part has its specific tRNA, and each tRNA has a specific amino acid. Now rRNA is ribosomal RNA, which is actually 80% of the total RNA. It has a two subunit, one is a smaller subunit and one is larger subunit. As you see, the ribosome actually has these subunits. You already read about in class 11th also and class 9th also. It synthesizes the protein only, because it read the mRNA and synthesize the protein. tRNA transfer and take in the amino acid and code the amino acid according to the mRNA codon. And mRNA is actually a 5% of the total RNA, which carry a genetic code from the DNA to outside. Taken the genetic codon from DNA to outside and is present in the form of a triplet. Now what is the structure of tRNA? Actually, this is the structure of tRNA, where one side is 3 to 5 and the 3 part is consist of a D loop, then lower part is anticodon. As you see, 3 codes are there, that's why it is called as a triplet. It only code the triplet type of the amino acid according to the codon. Variable arms is also there, T loop is also there. So how they code the amino acid? The code of the amino acid is according to the codons. Like amino acid is going to be code like methionine is going to be code from AUG. That is a start codon. If we go for the stop codon, UAA, UGA, UAG, these are the stop codons. So different type of the codon and triplets coded different type of the amino acids. Now the next one is RNA translation. Not so big, but also not so small. The mRNA which is formed is now transcribing the protein by the help of rRNA, but it has taken 3 steps. Number 1 step is initiation. This rRNA bind to the mRNA and start at the start point of mRNA, and this tRNA carry the methionine As you see, start codon actually bind the methionine met. So bind the methionine to start codon AUG. The codon is AUG present in the below part. As you see, the below part of tRNA, यहाँ पर this part AUG is present and it coats the protein called as methionine. The second one is elongation. Actually, amino acid taken by the tRNA is linked by the form of a chain. One amino acid, then another amino acid, then another amino acid, then another amino acid, and one by one it coat the whole chain. Now the termination, the finished polypeptide is released by the help of a stop codon, and the stop codon is UAA, UGA and UAG. Now the next part is lac operon. Actually, very simple. I will tell you it in a very simple way, but lac operon actually given by the scientists Jacob and Monod. It here the process is going to be explained in two terms, one in the presence of lactose and one in absence of lactose. So how can you find out? Firstly, think about it. In the absence of lactose, there is one I gene present in the lac operon which code the repressor. It can happen both in both cases. One in the presence of a lactose and another in absence of a lactose. Suppressor will code, means this repressor will code from IgM, but the repressor is going to bind with the operator part of the lac operon and block the way, and because of this RNA polymerase is not going to transcribe, or RNA polymerase is not going further to transcribe the mRNA and form the proteins. That's why RNA polymerase not go further and no protein is synthesized. This condition is called as the switch off technique. But in case if lac operon is present in lactose medium, I gene again form the repressor, but this repressor is binded by the molecules present in that sample of lactose, which bind the molecule of repressor, and because of that repressor will not occupy the place of an operator. Now operator part is open, the technique is switch on. RNA polymerase is not blocked by anything. RNA polymerase is going further and transcribe the 3 gene which is present in the lac operon. The 3 gene is actually Z gene, Y gene, A gene. This Z gene actually transcribe the protein called as beta galactosidase, which actually have a function of hydrolysis of disaccharide, means hydrolyze the disaccharide. Disaccharide is a type of a carbohydrate sugar. Now what is the work of Y gene? Y gene actually produce permeases which increase the permeability, which increase the capacity of a cell so that it transfer the beta galactosidase in the another part. And the third one is A, which transcribe the transacetylase, and transacetylase actually working in the part when glucose is broken down and form a pyruvic acid by the help of glycolysis, where it forms the acetyl coenzyme A, so it transfer the acetyl group to the pyruvic acid. These are the function of lac operon. So lac operon is also important, and the last of the moment genetic code is there. Given by George Gamow, total number of the codes are 4 की power 3 means 64. Start codon is AUG, code the methionine as you already know, and stop codon is UAA, UAG, UGA. The synthesis is actually of 20 amino acid, GGU, GGC, GGA and GGG. These 4 coded the one protein called as glycine. The CCU is a proline, and the protein tryptophan code by many amino acid. Now these are the chapters. You have to pause this video, write the notes, and you have to understood each and every task. Now the main part is PYQ. So let's go for the PYQ. The PYQ has some questions, but I will publish the main questions and answers in the next video tomorrow. So how is the length of DNA usually defined? First question is this. The second question is, What are the mention the number of bases or base pair in DNA in the bacteriophage 5 into 174 and in E. coli? Name the component of a nucleotide responsible for giving 5 is to 3 polarity to a polynucleotide. Now, where is nucleotide in the glycosidic bond present? Calculate the length of a DNA of the bacteriophage lambda, which has 48,502 base pair in its DNA. Name the scientist who proposed the central dogma. Central dogma is actually from DNA to RNA, from RNA to mRNA. Sorry, from DNA to mRNA and mRNA to protein. This is central dogma. Now the next one is write the features that a biomolecules must fulfill to able to act as a genetic material. And the last one, it is established that RNA is the first genetic material. Explain giving 3 reasons. All the answers must be published tomorrow, and many other important questions are published tomorrow. You have to hang with these questions. It's very important for your examination. So this is the total video of 21 minutes. Now you have to enjoy the video and like, share, subscribe, and also share this video to your someone. Best result will coming soon. Thank you. Have a nice night and goodbye.